Welcome to our genetics class. The most important thing you need to know about this class is that it's a flipped class. So that means you will watch the lectures before you come in, and then you'll have an opportunity to ask me questions about them. Or I may ask you to ask me a question, so it's good to think of something. The other thing is that you need to look at the chapter, take a look at the chapter, especially for those of you who learn more from reading than watching. The textbook is a great resource. By doing these things outside of class, it will free up time in class for us to do a group project, to discuss case studies, to have other special lectures, and of course to discuss the material. This has been very effective when I've been teaching so far, and I hope that you will enjoy this approach. You will be put into groups eventually, and those groups will work together developing a poster. This poster will be presented at the end of the semester, and by poster I mean a scientific poster, so it'll be like a mock scientific poster session. One of my jobs is to help you become a scientist or help you become a doctor or whatever your career plans are, but I want you to get a taste of what science is really like. All the details about the course are laid out in the syllabus, which you will be receiving or you probably have already looked at. The syllabus, of course, is subject to change, but it gives you an outline of what the course will be like. So once again, I want to welcome you to our genetics class, and I hope that we have a great semester. Now here comes the voiceover.
Genetics is fundamental. It plays a role in almost every aspect of biology. As you can see, the word has many associations. Words such as DNA, inheritance, proteins, functions, breeding, cells, etc. During this course, you will understand why so many of these words have been associated with genetics. Genetics has been a powerful tool that has given us insight into fundamental questions. For example, Mendelian genetics succeeded in explaining how traits were inherited and overturned previous explanations. And we will discuss this further in this presentation. Genetics is much more than a way of understanding why we look like our relatives. Today, genetics is used to explain how organisms function and how disease occurs. Heredity and fertility are ancient human concerns. Ancient cultures were fascinated with these subjects. They worshipped gods that they believed could influence their reproductive fate, but did not understand how these traits were transmitted. Genetics is not new, but our understanding has certainly changed over the years. Despite not knowing how genes could be selected for, humans in prehistory had much success in selective breeding of plants and animals for agriculture. This began in prehistoric times, around 12,000 years ago. Here is an image from about 10,000 years ago, and this piece of Syrian art depicts artificial pollination of plants. Animals were also domesticated. Here is the goat, Capra hircus, which was domesticated in the Near East as early as 11,000 years ago. And you can see during that time, other animals were domesticated, including sheep, pigs, and cattle. Today, we have continued to modify organisms to suit our agricultural needs. The most famous being genetically modified plants. And these have been produced through genetic engineering to resist herbicides, insect pests, or in the case depicted here, golden rice that biosynthesizes beta-carotene, a precursor of vitamin A. This is important because there are areas, particularly in Asia, where there's a shortage of dietary vitamin A. And this deficiency is estimated to kill 670,000 children under the age of 5 each year. Genetically modified insects, particularly mosquitoes, have also been produced. These are used to either reduce mosquito populations or to create mosquitoes that are resistant to diseases that affect human populations, such as dengue, fever, and malaria. Recently, a company called Oxitec, has developed a mosquito that carries a dominant lethal gene. So when males are released into the environment, this gene is activated and causes the progeny of the females that they mate with to die. And this is a way of knocking down populations without using insecticides. And this has been used in Brazil, in the Caribbean, and in fact, Oxytex has proposed doing studies right here in the Florida Keys. Returning to the history of genetics is important. And we want to know, how did we get from basically a very poor understanding of heredity to our current state where we can actually actively manipulate genes through biotechnology. So we're going to first return to the Greeks. The Greeks had the concept of Pangenesis, and this was a cause of debate between Hippocrates and Aristotle. In the Hippocratic treatise on the seed, published about 500 to 400 BC, specific particles or gemmules carry information from various parts of the body to the reproductive organs that then get passed to the embryo at the moment of conception. And this is the idea of pangenesis, which is an inheritance of acquired characteristics. It was highly influential and persisted until the 19th century. Aristotle rejected the above and proposed that both male and female contribute to offspring and that there is some sort of struggle between male and female contributions. Much later on, the idea of preformation took hold. In this idea, a tiny miniature adult, a homunculus, perfect in proportion, was proposed to exist inside a sperm or an egg. There were those that believed that the homunculus was in sperm and those that believed that the homunculus was in eggs. In this model, traits were only inherited from one parent. This goes against many common observations of how traits are passed on but from parents to children. But this was a popular concept in the 17th and 18th century. An idea of epigenesis stood against the homuncular view. And this was proposed by William Harvey in the 16th and 17th century. He was famous for discovering how blood circulated. His ideas were fashioned after Aristotle, and he proposed that an organism is derived from a substance present in the egg, which differentiates into adult structures during embryonic development. This was a critique of preformationism, would later form the basis for the Germ-Plasm Theory. Microscopy was developed in the 17th century. Using this tool, by the early 19th century, scientists began to recognize that living things are composed of cells. There are two types of cells, prokaryotes and eukaryotes. Prokaryotic cells do not have a nucleus. Their DNA is contained in a nucleoid, which is at the center of the cell and floats within the cytoplasm. Eukaryotic cells contain a nucleus, and inside the nucleus resides the DNA containing genetic information. Both eukaryotic and prokaryotic cells contain a cell membrane, but only some eukaryotic cells, like plant cells, contain a cell wall in addition to the cell membrane, which encases the cell. Robert Brown described the cell nucleus in 1833. Theodor Schwann and Mathis Schleiden in 1839 proposed the cell theory. The cell theory states that all life is composed of cells. Cells arise from pre-existing cells, and that the cell is the fundamental unit of structure and function in living organisms. This led in part to the development of the germ-plasm theory by August Weismann, who lived from 1834 to 1914. He did an experiment where he cut the tails of mice off for 22 consecutive generations and looked to see whether the tail length changed. The tail lengths of the descendants always remained long. This ended the notion of acquired inheritance. The germ-plasm theory states that cells and the reproductive organs carry a complete set of genetic information that is passed to gametes, and this theory still stands today. This brings us to the founder of modern genetics. Here, Gregory Mendel is memorialized on a stamp from the Vatican commemorating the 100th anniversary of his death. Mendel proposed a new theory of inheritance in his paper Experiments on Plant Hybridization, which was published in German in 1866. His ideas did not receive the attention they deserved until after his death, but since they had been rediscovered at the turn of the 20th century, his influence on biology has been immense. Mendel chose a very good model system to study how traits were transmitted during reproduction. He used pea plants that could either self-fertilize or be crossed to another plant. By following a number of traits through many generations of pea plants, he formulated his two laws of genetics. Mendel's first law, the law of segregation, states that each individual organism possesses two alleles encoding a trait. Alleles separate when gametes are formed, and these alleles separate in equal proportions. Mendel's second law of independent assortment states that alleles at different loci separate independently. So how does this work? Let's say there's an organism with two chromosomes that is depicted here. The chromosomes in red are from the mother, and the chromosomes in blue are from the father. During metaphase 1 of meiosis, these chromosomes will be in two arrangements depicted on the left and the right. After metaphase 2, this produces four types of gametes. This process of meiosis ensures that alleles at different loci separate independently, and we will be discussing this more in the next lecture. Ideas about genetics began to influence human health, and one of the great examples of this is hemophilia that has occurred in royal families. You can see that hemophilia is an issue in the pedigrees of both the British, Prussian, Russian, and Spanish royal families. Looking closer at this pedigree, you can see that the carriers are marked in red outline, and the affected individuals are marked in solid red. Looking at this, you can get a sense that it's rare, very rare, that a female is affected, and only approximately 50% of the male progeny get the disease. And this is associated with a sex-linked trait, and the gene that causes hemophilia is on the X chromosome. And we will be discussing sex-linked traits and sex determination and the like in future lectures. In the 20th century, genetics began to really flourish. Walter Sutton proposed in 1902 that genes are located on the chromosome. Thomas Hunt Morgan worked with fruit flies and identified the first genetic mutant in 1910, and this was the beginning of transmission genetics. Ronald Fisher, John Haldane, and Sewall Wright laid the foundation for population genetics, and their work still influences research today. James Watson and Francis Crick solved the 3D structure of DNA in 1953, using data from Rosalind Franklin, and this was the beginning of molecular genetics. By the 70s, the first recombinant DNA experiments were performed, and these revolutionized genetic research and allowed for cloning, genetic engineering, and many other techniques that are used today in biotechnology. By 1977, DNA sequencing methods were developed by Frederick Sanger and Walter Gilbert. This allowed us to decode the sequence of genes for the first time. The central dogma of molecular biology was becoming more clear. DNA is transcribed into mRNA, which is then translated at the ribosome to become proteins. And proteins do the majority of functions in the cell. Other techniques helped advance molecular genetics, molecular biology, and such techniques such as the polymerase chain reaction, which was developed by Kary Mullis in 1983, allowed for the rapid amplification of DNA, which was incredibly useful in things such as cloning or the detection of viruses. There's many, many applications of PCR. The community began to realize that to really understand the biology of organisms, we needed, and especially ourselves, we needed to develop genomes. A genome contains the complete sequence of DNA in the nucleus. And this would contain the sequences of all the genes and all the sequences that regulate those genes. And understanding this information is a key to understanding the genetics of any organism. So in 1990, the Human Genome Project was launched. At the same time, genome projects were launched for simpler organisms, such that by 1995, the first complete genome of a living organism, Haemophilus influenzae, was determined. Now that we're in the genomic era, many, many species have had their genomes sequenced. And I chose these particular species because they represent the kind of diversity of genomes that have been produced. Here I'm highlighting E. coli, which is, of course, a prokaryote. It has a relatively small genome. It is a single-celled organism, and its DNA is not packaged into chromosomes. And overall, prokaryotes are a little less complex than eukaryotes. So let's focus on the eukaryotes for a moment. Saccharomyces cerevisiae, a baker's yeast, and this is a single-cell organism. Arabidopsis thaliana, which is the thale cress plant. C. elegans, a nematode, a small roundworm. Drosophila melanogaster, which is a fly. Aedes aegypti, which is a mosquito that causes dengue fever, Zika, and chikungunya, which you may have heard about. It actually exists here in Miami. Musculus, the house mouse, which is one of the main workhorses of vertebrate genetics. And of course, us, Homo sapiens. Now, when you look at these genomes, you notice that the multicellular eukaryotes have a greater number of genes than the single-celled yeast, Saccharomyces cerevisiae. You can also notice that among the multicellular eukaryotes, even though we have a plant, different invertebrates, which are the C. elegans, Drosophila melanogaster, and Aedes aegypti, and two vertebrates, the mouse and human, we see a relatively similar number of genes. The same cannot be said for the size of their genome. So there are, of course, differences, but it suggests that genes are, for the most part, conserved through evolution. This is clear in the comparison between Drosophila melanogaster and Aedes aegypti. Both are insects, and they both are dipterans, and dipterans simply means that they have two wings. And you can see, although they have a basically similar number of genes, Aedes aegypti has a much larger genome. And why that is still a mystery, but I think it's something that is very interesting. When we look at the vertebrate genomes I'm showing here, the human and the mouse genomes, we see that they have a very similar number of genes. And actually, the overall size of their genome is not that different, which I think was quite a surprise to people, especially given that mice and humans look and behave in very, very different ways. So it seems that it's likely that the regulatory regions in these genomes are critical and influencing how genes are expressed to cause changes in these organisms to make them different from each other. And we will be discussing this later in the class. Obviously, genetics has influenced research, especially in the age of molecular biology. Genetics has influenced, obviously, developmental genetics, molecular genetics, transmission genetics, population genetics, the genetics of viruses, microbes, plants, humans, and other animals, and also fungus. And using genetic approaches, we've been able to understand a lot more about these diverse organisms. One of the ways that we understand biology in the laboratory is using genetically accessible model organisms. And what do I mean by genetically accessible model organisms? These are organisms that we can genetically modify in the lab. So I'm going to give you a list of these model organisms and tell you a little bit about them. So there's Saccharomyces cerevisiae, which is the baker's yeast. Here we're showing this yeast dividing and budding. This organism has been very important in understanding the cell cycle, and I think that's been one of its greatest contribution. Also understanding just the basics about how transcription and translation work. C. elegans, the nematode, has also made many great contributions to developmental genetics, trying to figure out how an embryo goes from one cell to many. It has even made an impact in neuroscience and behavior. And one of the great things about this organism is that it has a very short life cycle, so which is great for genetics. And it's also a hermaphrodite, so you can self these organisms, that these organisms actually contain both sperm and egg inside of them. Although it is possible to do traditional male-female crosses, or in this case, male hermaphrodite crosses, because occasionally you can get males in C. elegans. Drosophila melanogaster is probably the most well-studied model organism ever, and certainly the best of all the invertebrate model organisms. It has been worked on since Thomas Hunt Morgan started his work in 1910. There's a huge collection of mutants, and you can study many processes, including developmental genetics, transcription, translation, anything you want to do. They have interesting behaviors, and increasingly it has become an important model for neuroscience and behavior. There are also vertebrate model organisms. One of the newest vertebrate model organisms is a zebrafish, Danio rario. And this model is particularly exciting because, especially during embryogenesis, these fish are transparent. So you can do all sorts of cell imaging techniques to look at the effects of different conditions or loss of genes on individual cell behavior. It is also a great model in which you can manipulate genes using forward genetics, which is also true of C. elegans and the fly. And we will discuss the concept of forward genetics in the future. I just wanted to put that out there for you. And here we have the mouse, Mus musculus, the house mouse. This is a very important model organism that has allowed us to connect different genes with diseases, and a lot of these genes have actually been conserved in humans. The mouse has definitely been the most extensively studied vertebrate model organism, and it is of incredible value. In some ways, it's one of the best models to study immunology because they have a very complete adaptive immune system that is somewhat similar to ours. They have interesting behaviors, and also some people have actually studied development in the mouse. The most popular plant model organism is Arabidopsis thaliana. This model has allowed us to understand many details about how a seed becomes an adult plant, the growth of roots, the response to light, all sorts of things. So, and this work in Arabidopsis has been applied to other plant models like maize. From my point of view, model organisms have driven genetics research. We need these model organisms because there are limits to the experiments that can be done on humans, obviously. We can't conduct artificial breeding experiments with humans. We can't do any invasive experiments that would damage human health. So we need organisms where we can really understand gene function. We tend to choose organisms that have a short generation time as model organisms. And one of the advantages of Drosophila, which its life cycle is depicted on the left, is that you can go from an adult to an egg and back to an adult again in just 10 days, which is really good. Also, for those interested in developmental genetics, an egg can completely develop in 24 hours into a first instar larva, which is depicted here. From this research with model organisms, we have discovered that many genes are conserved from yeast to humans. And this is great because we can do experiments in these organisms and then test hypotheses in humans about how certain genes may be affecting human health. A great example of the conservation of genes is the PAX6 gene. This transcription factor has an effect on eye development from flies to zebrafish to mouse and even to humans. So you can see here when a fly, and here this is Drosophila on the right, loses this gene called eyes absent, which is the ortholog, or which means basically the same gene but in a different organism of PAX6, you see a near total loss of the eye. Loss of PAX6 in vertebrates all cause eye defects, including decreased eye size, defects in the lenses, the retina can be disturbed and malformed. And an interesting thing to notice is that even in humans that contain a mutation in PAX6, if you just lose one copy of the PAX6 gene, you can get defects, and which are pretty serious and likely, more than likely, obviously lead to blindness. So this is an example of a dominant gene, and we will be talking about that in later lectures. The work that has been done in these model organisms has led to many discoveries about human health. And here I'm just listing the linkage of a number of diseases that have been linked to genes on human chromosomes, such as hemophilia, which we discussed earlier, different immune deficiencies, breast cancer, polycystic kidney disease, Tay-Sachs disease, Alzheimer's, cystic fibrosis. So genetics has really made an impact, and I think it's just really important for us to really try to understand genetics this semester, because I think genetics is going to change things in your lifetime and open up new doors. So I just want to review a few key concepts in genetics. I imagine some of these concepts have come through in the lecture, but I just want to stress them here. The first one is that the gene is the fundamental unit of heredity. Genes are inherited. A complete set of genetic instruction for any organism is in its genome. The genome dictates how an embryo forms, influences behavior. There's a whole range of things that the genome is doing. The genetic information contained in an individual is its genotype. And this, of course, is contained in the nucleus. The traits that you see, for example, having red hair or blue eyes or darkly pigmented skin, all of these things are the phenotype of the gene that a person has inherited. Genes come in multiple forms called alleles. So you have a gene, it comes in different kind of flavors, and those flavors are alleles. Some people have, for example, in the example of PAX6, some people have a wild type copy of PAX6, and then some people also have a mutant copy of PAX6. And these would both be alleles of the same gene. Genes are located on the chromosome, which is composed of nucleic acids and proteins. The number of chromosomes can vary with species. Humans, for example, have 46 chromosomes, and very simple prokaryotes like bacteria only have 1. The genetic material that genes are made of may be DNA or RNA, but for the most part, DNA is the main source of genetic information in most organisms. Well, I can't stress to you how fundamental genetics is. I've done my best during this lecture to try to do that. It forms the basis for so many things, and in the next lecture, we're going to discuss how genetic information is transmitted, both in dividing cells during mitosis and in the production of sperm and egg, a special form of cell division that is called meiosis. So I hope you enjoyed the lecture, and I'll see you next time.